The Autism Research Institute depends on the generosity of its donors, like you, to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
This is a rapidly evolving field, but I'll say in the outset that really over the last probably 6 months and through the facilitation offered by the two major autism groups that I've been affiliated with, so Autism Research Institute and Autism Speaks, it's been clear that we can distinguish primary mitochondrial disease and secondary mitochondrial dysfunction, and it really, the vast majority of children with autism spectrum disorders will not have a primary mitochondrial disease defect. It may only affect 1 to 4% of children with with autism spectrum disorders. And the remaining may have ecogenetic forms of mitochondrial dysfunction. I'll say that word again, ecogenetic, because I'm going to finish my talk with that word and how what the implications for the interaction between our ancestrally transmitted gene set that we've received from our ancestors who had to interact with their environment, the foods in their environment, and the microbes in their environment, the viruses and bacteria and fungi in their environment. And as you might imagine, only those individuals survived to reproduce that had the genetic capacity to handle the changes that occurred in the environment and the various infectious agents that existed in where our ancestors originated. So many years ago, I put together a slide just to indicate all the different medical manifestations that can occur with primary forms of mitochondrial disease. And they can range from muscular dystrophy clinics, epilepsy clinics, leukodystrophy, bipolar depression, schizophrenia, sudden infant death. But we're really going to be focusing, on autism spectrum disorders here. So, in the beginning, there was a honeybee. Okay, and this honeybee was absolutely essential for maintaining the entire health of an ecosystem. That honeybee is not unlike mitochondria within our cells, and our cells are the ecosystem, and mitochondria are constantly, measuring the chemistry and the physical environment in which they reside, the oxygen content, the temperature, the pH, the presence of various nutrients. And in response to that, they actually send out signals continuously to the nucleus, to peroxisomes, to other cells. They're constantly communicating, updating, neighboring cells, organelles within the cell in which they reside. And for about 50 years, mitochondrial specialists or biochemists have been cracking open cells and pulling out the mitochondria and studying them like a little honeybee in a glass flask, where we can identify all the different proteins in a mitochondrion, identify over 500 different functions that they perform. But until we actually evaluate their function in the context of the whole cell and then ultimately in the whole individual, we do not understand the function of mitochondria. Okay, it's much more than just oxygen consumption and ATP production, and I'll go through some of those things. Okay, so mitochondria create the words, the chemicals that cells use to communicate. Mitochondria sense and regulate the internal environment of the cell. They adapt the cell to the changing environmental conditions. The cells need about 1,500 different genes that are specifically targeted to mitochondria for mitochondrial biogenesis. There is no such thing as a single mitochondrion. They are constantly fusing and fissioning from one another. Okay? 1 in 2,000 children born will develop definite forms of mitochondrial disease in their lifetimes. We'll call those the primary forms of mitochondrial dysfunction. About 10 to 20% of those definite forms may have an endophenotype that qualifies for a diagnosis of autism spectrum disorder, but only 1 to 4% of all ASDs will qualify as a primary mitochondrial disorder. And that means that really 96 to 99% are not primary mitochondrial disorders. Why is it? And then mitochondrial biology is is what I call the secret life of mitochondria. In my view, the cellular defense functions of mitochondria were actually more ancient. With the first eukaryogenic event that occurred dating back to as far back as 2.5 billion years ago, or as recently as 0.9 billion years ago, a single cell free-living bacterium that consumed oxygen was fused with a archaebacterium to produce the first single-cell eukaryotic cell. So the Precambrian seas were just awash with the bacteria and viruses, and then eventually with single-cell eukaryotes. And in that time, the only single-cell eukaryotes that could pass on their genes to the next generation were the ones that could survive this complex microbial ecosystem in the oceans, that be able to survive the viral infections, survive the parasitic viral infections. Okay, and for single cells can do just fine on glycolysis. They don't need mitochondria for energy production. Okay, you just don't need it. It's really you need mitochondria for energy production when you start putting multiple cells together to make animals and plants. Okay, so with the Cambrian explosion, that's when the real hidden value of mitochondria really became apparent, and it was so powerful. It is now its remnants are all everything that we see today in this in our living world. So mitochondria produce the first danger signals, and let's see. So some of these. Some functions I'll just briefly go over. Oxygen consumption is an obligate function of mitochondria, but ATP synthesis is an inducible function. It is not something they have to do all the time. Okay, mitochondria can regulate their ATP production through uncoupling proteins. They can regulate it through a variety of different other mechanisms, including nitric oxide synthesis. So they don't have to make ATP. Folate, B12, S-N-acetylmethionine, methionine, cysteine, glutathione metabolism, all are regulated in part through mitochondrial metabolism. Cellular redox, which is perhaps the major determinant of intracellular metabolism, is fundamentally controlled by mitochondria. I'll say that perhaps the two things that influence redox more than anything else are basically our diet nutrient intake and our exercise level. Okay, and then after that, it's the mitochondria that work with that. Crosstalk with peroxisomes, glycolate and oxalate metabolism, the work capacity of the cell, and their growth capacity, and their decision to commit cellular suicide, apoptosis. During cellular development, we have to prune out a lot of neurons and synaptic connections. We have to prune out a lot of cells, and mitochondria are intimately involved in that, that pruning process. Nucleotide synthesis. So actually, the four-step in de novo pyrimidine biosynthesis is dihydroorotate dehydrogenase, and that's has to send electrons to the mitochondrial intermembrane electron transport chain, and that's responsible for making all the U's, T's, and C's of the cell. But you already know about ATP, but GTP is also made in mitochondria. And these nucleotides don't just have energy-carrying characteristics. You know that they're informational molecules once they're polymerized into DNA and RNA, but it turns out they're also informational molecules as monomers extracellularly. So once extracellular ATP, once cells actually will produce extracellular ATP, and it binds to receptors, and I'll talk to that in the rest of the talk. So cholesterol, cortisol, steroid hormone synthesis, vitamin D activation, inactivation, porphyrin iron cluster synthesis, heme biosynthesis, calcium, iron, copper metabolism, and the first committed steps of innate immunity. All these are mitochondrial functions. Oh, and the one that frequently gets omitted is meiotic recombination. So if you take a baker's yeast, Saccharomyces cerevisiae, and you make a row 0 cell, pull out the mitochondrial DNA, one of the most profound defects that you observe is that now those cells cannot sporulate. They cannot produce gametes. Okay. It also seems to be that, at the heart of why a number of hybrid crosses, for example, between a horse and a donkey, give rise to a mule that is sterile because of the incompatibility of the mitochondria carried from a horse, with the nuclear DNA, carried from the donkey. Okay, there are lots of examples of these things that are kind of arcane, but fun. All right. So remember, there are always two ways that the cell can produce energy. The most ancient is glycolysis, which is ATP production, and let me go to this one. So I'll start. So ATP production in the absence of oxygen, using glucose, and basically, so with one glucose, with 6 carbons of glucose, you can make 2 ATPs net and a lactate and 2 lactates. Okay, that also happens when we injure ourselves. If we sprain our ankle and our cells start swelling, we shift our metabolism. Our cells shift from an aerobic metabolism to a glycolytic metabolism. It happens after strokes and MIs, and that turns out that metabolic shift and then the restoration of aerobic metabolism after that shift is made, that is associated with healing, is an important part of regenerative medicine biology. Another interesting thing about glycolysis, it's highly inducible. Glycolysis can be induced 1,000-fold over baseline levels. If you take Usain Bolt at the starting line of the Olympic 100-meter dash, measure his glycolytic activity at that time, you can call it 1. At the end of a 100-meter dash, he's working at a level of 1,000. Was that me? What was? Oh. Anyway, that's So congratulations to Usain. But anyway, so you can rapidly induce glycolytic ATP production. Now, what about this other method that's so important of using oxygen to produce ATP? This is more like the hare in the tortoise, as in the tortoise in the hare race. Okay, aerobic metabolism and the production of ATP is about 50 times slower. It runs about 2% of the capacity of glycolytic energy metabolism. But it turns out that speed of ATP production is not critical. It's the efficiency. When you go through a winter and calories are restricted, the people that survive at the end of that winter are the people that were able to eke out the very last amount of ATP from what they had stored in terms of their fat reserves. Okay, and then when you exercise, we can only induce the aerobic metabolism of mitochondria about 2 to 3-fold. Okay, so it's not 1,000-fold inducible; it's 2 to 3-fold inducible. Nonetheless, there are a lot of other roles that it plays, and we go through that. So bump, 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 bump. 2 ways to make make energy. Okay, mitochondria are made from about 1,500 genes. That means that the proteins involved, in mitochondrial biogenesis, are spread all throughout the 23 chromosomes. There are only 13 proteins that are made and are encoded by mitochondrial DNA. All the others are made in the nucleus, and as such are all subject to tissue-specific gene expression. So the genes expressed in the nucleus for mitochondria in a retinal cone cell are completely different than, well, not completely different, but very different from the genes expressed in the nucleus of a liver cell or a kidney epithelial cell or a neuron of the CA1 region of the hippocampus, or in white cells. Okay. So each one of those 1,500 genes can be considered to be controlled by something akin to an audio mixer where every channel can go up and down according to different programs. And these genes are, again, come from different chromosomes. The mitochondria perform about 500 different mitochondrial functions. And the genes are regulated coordinately for some sets of genes and discoordinately for others. You can't, make predictions about, this person had increased mitochondrial function or decreased mitochondrial function. That kind of loses meaning when you got 500 mitochondrial functions to talk about. All right, so 500 different mitochondria. So in the human body plan, we have about 500 terminally differentiated cell types. There is a different mitochondrion specialized for every one of those cells. Okay, so they are adapted to meet the metabolic demands of each individual cell type. So if I had a pointer, I'd point out some of these different mitochondria here that come from on the upper left. This is another thing. When we crack open cells and pull out mitochondria, what happens? Mitochondria normally are present in filaments, but when we snap them apart, pull them out of the cell, they snap into like little spheres according to soap bubble physics. So you lose their normal configuration when you crack open the cell as well. And what we've learned is that actually dramatically influences their function. Their function when they're in long filaments and networks within a cell are very different from when they're just in little balls. And we are always measuring just the function in little balls. Okay, and so this is what we'll call the spaghetti and meatball transformation between mitochondria. Okay, so just below that top left slide is a slide that also has some spherical mitochondria, but if you note the cristae inside, they're much more sparse. Those are the mitochondria within a primary oocyte. Okay, so an egg, and an egg will contain anywhere from 200,000 to 2 million of these little balls, and each one of these little balls will have about 1 mitochondrial DNA. It's a little bit different than the normal situation, okay, where mitochondrial, will usually be associated in nucleoids with 5 to 7 mitochondrial DNAs at a minimum. Okay, well, I won't spend too much more time on this. I'll go through. So, on the right. Okay, so on the right we have a line and dot model of intermediary metabolism. And that's just to remind me to tell you that, first of all, not every cell does every reaction that is present on this, on that diagram on the right. Part of the metazoan, solution to complexity was specialize. So when certain cells, differentiate to become neurons or retinal cells or pancreatic beta islet cells, they give up certain metabolic reactions so that they can do others better. Okay, this is something that Adam Smith, identified and was the basis of the Industrial Revolution. It's something that Henry T. Ford, brought to commercial success in creating the first assembly line, where each component was of the car, of the Model T Ford, was produced by a specialist in wheels and frame and engine, lights, and none of those people could actually put together the whole car. Okay, but together they worked to produce something that had never been made before. Okay, so that's where our mitochondria are helping out. So mitochondria don't do all these things in all the different cells, but they do 90% of the catabolic reactions, and 70% of the anabolic reactions pass through mitochondria at some point. All right. So mitochondria also adapt to genetic, and environmental changes. So you can have a primary defect. So these, if you, imagine a few of the mitochondrial functions, I may have about 30 of mitochondrial functions, imaged here. And let's say we inherit a defect that produces, an abnormality in one. Well, that produces a cascade of compensatory abnormalities that result in decreases and increases in certain functions. And finally, we have some function that is, related to a high-order symptom that we might see that might be seizures, or might be, the susceptibility of the gut to particular allergens. Okay, so or immunoreactivity, the overstimulation. So And that turns out to be the actual cause of the symptom, but in certain mitochondrial disease, you'll have a primary defect, or you can imagine that could be caused by some environmental, exposure, okay, some environmental agents. All right, so the treatments, that's effective is, directing at the proximate cause of the symptoms in the secondary disorder. Many of the primary defects and genetic disorders are not really amenable to treatment. Okay, you can have an increase. For example, you can duplicate, chromosome 15q11, and that'll produce another cascade of, effects. Okay, and you can measure all those, but that can produce also symptoms that were the same as that were produced by the first set of abnormalities as I showed, and so you can have a treatment that actually can be effective for some a symptom that is caused by several different things. Okay. All right. Mitochondria are dynamic. They're continuously fusing and fissioning. So on the right, you'll see a green mitochondrion that will fuse and exchange its contents with the red-marked mitochondria. You can follow this green one again. It's going to go ahead and touch, and we call it the kiss and run. They'll fuse and then they'll fission. They share mitochondrial DNA. They share proteins with one another. That's continuously going on. When cells are exposed to progressive gradients of stress, what happens is you go from most of the filamentous or tubular, you see down in B, at this panel in B, mostly tubular mitochondria. As you increasingly expose the cells to increasing magnitudes of stress or metabolic deprivation, nutrient deprivation, heat shock, pH shock, oxygen shock, what will happen is you progress to more and more spherical mitochondria. Okay? So the mitochondria fragment. And interestingly, one of the first things you can see when a cell commits to apoptosis structurally are fragmented mitochondria. Okay, that happens even before the flip of the phosphatidylserine from the inner leaflet of the plasma membrane to the outer leaflet of the plasma membrane that is detected by a Nexin-5, a common marker of apoptosis. All right, the language of metabolism is, the language of the cell is metabolism. Now this is a very busy slide, and I really wish I had a pointer. But let's start at the top, and I'll just walk you through. So the substrates that this cell will consume are glucose and lipids. And those are actually processed by mitochondria that are specialized for carbohydrate metabolism or fatty acid metabolism. Mitochondria are then producing, so if you look at the left-hand mitochondrion, it produces mitochondrial DNA under conditions of stress. It will release mitochondrial DNA and it will fragment and release pieces of itself and peptides that are actually inflammatory. So that's what the little flame is there, is to indicate inflammation. And so those, and it's by virtue of the fact that mitochondrial proteins frequently start with a formylated methionine, okay, just like bacteria, that pieces of it can be recognized, as foreign and will activate the early innate immune responses and will actually recruit phagocytes. Okay, so on the other, go a little bit farther down there, you'll see a little graph that says EATP. So there's a happiness point, and I'll call this. So there's the principle of metabolic matching that I'd like to develop. And when a cell is in an environment that meets all of its nutritional and physical needs, mitochondria will produce a set of small molecules that include things like ATP, but also include things like alpha-ketoglutarate, succinate, and other molecules that get sent to the nucleus and will get sent outside the cell that participate in, that actually create a little halo around the cell of high concentration, and that halo glows with a color that says, "I'm well." Okay. And when the cell finds itself in a hostile environment, when it's exposed to any environmental chemical, whether it's mercury, cadmium, or this is polybrominated diphenyl ethers as flame retardants. You will produce a change in the metabolic function of the mitochondria and will change the small molecule words that they use to communicate with the nucleus and neighboring cells. So anyway, the principle of metabolic matching determines the dialogue that cells use to talk to one another, and will be the early alarm system in any exposure to either a viral infection, bacterial infection, fungal infection, or changes in nutrition or micronutrients. All right. So there are two complementary and conflicting states of mitochondrial function. In dividing cells on the left, what you have is non-dividing cells on the left. What you have are the classically understood mitochondrial functions of taking glucose or food, which is really sources of electrons, and converting those, burning those down to CO2 and water. So you're taking carbon skeletons, burning them to CO2 and water, and making ATP. So that has a name called anaplerotic function, so anaplerosis, and I'll go into that a little bit later. But in the next, so on the right-hand side we have cells that are dividing. So these cells need to not completely burn glucose to CO2 and water. Growing cells have to double their biomass every day. They have to make more phospholipid. They have to make more nucleotides. They have to make more protein every day. Okay, and that requires carbon skeletons. So if you're taking your carbon skeletons and burning them completely to CO2 and water, that's the end of cell growth. Okay, you can't do it. All right. You need a different function of mitochondria, and that's called their cataplerotic function. That's where substrates, foods will come into intermediary metabolism, pass through a step or two of the Krebs cycle, and then come out as a carbon skeleton that can be used for amino acid synthesis, nucleotide synthesis, and fatty acid synthesis. So the evolutionary origins of this, I'll just briefly go over it. So our ancestors had to deal with seasonal differences in availability of calories. In the winter, calories were limiting. That's on the right-hand column here. So calories were limiting. You had to work hard in order to get adequate calories. Okay, and there are certain hormones that were associated with low-calorie existence. I won't go too much into that, other than to go down to the line that said glycolysis. Okay, well, glycolysis during the winter is really low. This is the time when the tortoise wins out. Okay, the tortoise is making the best use of available carbon skeletons in the form of fatty acid depots, adipose depots. Okay, and that's how you make it through. In addition, you upregulate autophagy. Okay, so autophagy is much upregulated during our ancestral winter. One of the problems of human disease now is we're in an eternal summer, an endless summer. Okay, we don't have cycles of nutrients anymore. Okay, through winter and summer, and so we have now. I'll flip to the next, to the first column, where during the summer or in the daytime, calories are plentiful. You don't have to work so hard for your calories anymore. And glycolysis under those conditions is high, and mitochondrial oxidative phosphorylation is low, and cataplerosis is very high. So you're actually in a cell growth state. Okay, and cell growth manifests in adults as obesity, and in children it can produce. I'll introduce another term called developmental dyschronia. Because of the correlated growth requirements of embryogenesis, it is necessary to actually have cells move along developmental trajectories together, so that when one neuron gets to one place or sends out a projection, another neuron's ready to make the catch. It's kind of like a trapeze, trapeze artist. And if the catcher doesn't, is not at the right place. We do have something that's been here. Okay, thank you very much. That you're not going to make the catch, and those neuronal projections are going to fail. Okay, so anyway, so that's kind of a little brief overview about the summer and winter functions of mitochondria that are played out in another scale during the day when we eat and the night when we don't. There are smaller variations in that. And that's also associated with small variations, circadian variations in NAD and NADH, and as well as circadian variations in cholesterol biosynthesis, etc. So I'll leave it at that. All right. So anaplerosis. So that's Hans Krebs on the left. Well, yeah, so Hans Krebs is on the left in the 1940s. He worked out this cycle after working out the urea cycle, interestingly, first. Okay, so he did the urea cycle also. And he studied the flight muscles of pigeons. Okay, so these are pigeons have dark meat, okay, as opposed to your chickens and turkeys that have white meat. So white meat are fast twitch fibers that are for rapid burst glycolysis. So evolution placed fast twitch fibers in those animals in order to help them fly away from the fox. Okay, whereas migratory birds, like ducks and pigeons, use this form of slow twitch fibers that used anaplerotic intermediary metabolism, where you take things like pyruvate and fatty acids, aspartate and glutamate and branched chain amino acids, and you burn them all to CO2 and water, making ATP. Cataplerosis is the complement. This is what's used in for actually taking in a few things and then spitting them out. So methylmalonyl CoA gets spit out through succinyl CoA to make heme. You can also use pyruvate to make glucose through gluconeogenesis. Anyway, so the main theme is in anaplerosis, you're actually consuming like a great. I hesitate to use the term tornado at this particular point, but like a great vortex that pulls things in, and then in the case of mitochondria, will burn them to CO2 and water. And cataplerosis is where you actually take things in, and then you provide building blocks for cell growth. Okay, mitochondria can. So I told you before that so ATP is an inducible function of mitochondria. It occurs at the point of the fifth complex in the mitochondrial electron transport chain. This is a rotary catalyst. It actually turns in order to make ATP. The energy that is used to make ATP is stored as a proton motive force in this inner mitochondrial membrane space, and it normally would flow down through a proton pore in this channel. Okay, and we can our cells can actually regulate the amount of ATP by regulating the amount of uncoupling protein that will actually disperse this proton motive force, so UCPs, and there are other mechanisms too that will downshift mitochondria and allow them not to produce as much ATP. But under injury conditions, you can even have mitochondria that will consume ATP. Okay, so mitochondria, I mentioned, once you have cellular damage after a stroke, a heart attack, or a sprained ankle, or a cut in a wound, will shift their metabolism from aerobic metabolism to glycolytic metabolism. Okay, that's actually really quite, adaptive, because if you've severed your blood supply and you can't get very much oxygen, you only have a little bit of glucose coming in from collateral vessels, it's very useful to be able to make enough energy to survive without consuming oxygen, and that's done by glycolysis. Okay, but those damaged mitochondria can actually take ATP made from glycolysis and import it into the organelle and start turning the ATP synthase in the opposite direction, okay, to pump protons back in to the inner membrane space, okay, to maintain the mitochondrial membrane potential, to maintain the charge in the membrane. Why is that important? Okay, why? So it's important because the way that mitochondria tell the nucleus that they need supplies is by keeping the porch light on. Okay, so the porch light. Right? So by maintaining the charge, so remember there are 1,500 different proteins that have to get imported into mitochondria. Okay, how do they find mitochondria? They find them according to the charge on the mitochondrial membrane. Okay, so even if, so I'll just say, only charged mitochondria are resupplied. When mitochondria turn out the lights, when the membrane potential goes to 0, they don't get any new protein. Okay? So, and then they're essential for de novo permanent biosynthesis. I already mentioned that. This enzyme dihydroorotate dehydrogenase on the intermitochondrial membrane produces uric acid that then is a substrate for uridine monophosphate synthase, or UMP. And then that UMP is then a source for all the T's and the C's, that are used for RNA, DNA synthesis, and for carbohydrate activation for glycogen metabolism. All right. So here are some many faces of mitochondrial disease. There are over 300 genetically distinct forms of mitochondrial disease. Each one of these has a little bit different mitochondrial disease. It's common for mitochondrial disease children to be, normal at birth. Here you can see the little girls in their Easter outfits. Both those girls actually had a disease called Alpers syndrome. And one of the things that each one of these children, even though they all have different disorders, different primary causes, the thing that connects them is none of them survived past 10 years of life. Okay, so I'm going to skip to the chase here and say that basically the incidence of mitochondrial disease is 1 in 2,000 live births, but only half of those will present before the age of 10. So that's 1 in 4,000, and half of them will present with symptoms after the age of 10. How do you diagnose it? We have something called the modified Walker criteria. We use a gradient of disease severity to define the categories of mitochondrial disease, and definite mitochondrial disease criteria have been selected to help us identify primary forms of mitochondrial disease. These are monogenic forms typically, genetic forms. It allows us to provide family counseling for the patients, and in some cases allows the development of specific treatments. So I'll bring out this last little square that first of all, it requires for a definite diagnosis, you need either DNA or a muscle biopsy. And in the muscle biopsy, the level of electron transport chain deficiency has to be 20% or less. Okay, it's being changed a little bit now, but I'll leave it at this for now. That's not what we normally see in children with autism, who usually will have, if they have mitochondrial dysfunction, they will have higher residual levels of mitochondrial proteins, and in some cases we'll actually have increases in mitochondrial proteins. So mitochondrial disease and autism are not the same. So children with mitochondrial disease are present. They will, in about 1 in 4,000 children will present before the age of 10. They typically have decreased hearing, decreased sensation to touch, decreased movement, decreased immune response, multi-organ dysfunction, and catastrophic setbacks, and often with infection, when progressive disease. Autism doesn't share many of those except for in select cases. Okay, so autism is really a triad of disorders involving neurodevelopment, brain, GI tract. Oh yeah, so I should make that point right away. So from an evolutionary point of view, there's an argument to be made that our brain is actually our second brain, and what we during the Cambrian and the development of the first multicellular organisms. Imagine the sea anemone. Sea anemone has a GI tract that is coordinated by the equivalent of a myenteric plexus, in which where the coordination of its GI tract was regulated by neurons and by actual neurotransmitters and peptides that eventually became co-opted with evolution to a site of greater neuronal development in what we call today our brain. Okay, but the gut, from an evolutionary point of view, was our first brain. And our brain is really our second brain, and that's why there's so many connections between the two, is because they're father and son. Okay. All right. Okay, so mitochondrial dysfunction in autism spectrum disorders. This is a really nice review by Dan Rosignol and Richard Fry, who really did a fantastic job of kind of culling together the papers that looked at mitochondrial disease and autism. And in this paper there was a 5% 5% of the children with autism spectrum disorders were characterized as having definite mitochondrial disease. Okay, that's a bit on the high side, and I'll show you why that. So then the flip side of that is 95% did not have a purely genetic mitochondrial disease. Okay, so but the problem with both autism and mitochondrial disease is there's an irreducible clinical heterogeneity. Okay, it's very difficult to do a clinical trial with relatively small numbers of subjects, because if you met one child with autism, you met one child with autism. If you met one child with mitochondrial disease, you met one child with mitochondrial disease. Okay, so this kind of brings up the point of elevated citrate synthase. I'll make a point of this. Citrate synthase is only found in mitochondria. It's a marker of mitochondrial mass. Okay, when mitochondria have primary defects, frequently there's a strong compensatory effort to increase the amount of mitochondria. Okay, so 44, or nearly half of the children with primary forms of mitochondrial disease, had a compensatory increase in mitochondrial function, and about a quarter of children with ASD. And I think I'm not. So I'll make a brief quick. Lactate is frequently used, and lactate pyruvate ratios are frequently used. But one of the difficulties of this is particularly pronounced in children with autism, is they don't like to have their blood drawn. Okay, and they will squirm and fight, and and one of the things that we really have a problem with is very little muscle activity in a child is sufficient to raise blood lactate significantly. Okay. Now, it turns out that can actually be used as a stress test, because if you have primary mitochondrial disease, it takes a lower amount of stress to raise the lactate, but it's still a complicated thing to interpret. Okay. All right. And so only about half of the children with real mitochondrial disease have elevated lactates, but 3/4 of the children with ASD and mitochondrial disease, had elevations. And I'll leave it there. Okay, so I made a point about the epidemiology of mitochondrial disease, and talking to you about that about 1 in 4,000 children will have definite mitochondrial disease. And I'll point out that it's actually mathematically impossible for 1 in 4,000 of children to have known mitochondrial disease, and 4% of the children with autism to have mitochondrial disease. Okay, so I'll just run through this. So if you take an imaginary population of 100,000 children and take 1% of those as having autism, well, let's see if I can do it from here. Okay, so the assumption is 1%, and everybody agrees with that. That's not that's not something that's in dispute. Okay, 1% of children will have autism spectrum disorder. So that means that 1,000 out of 100,000 children will have ASD. Okay, so ASD. And then if you take the assumption that the prevalence of mitochondrial disease within autism is 4.2%, as suggested by a paper by Olivieri, which is really one of the first that kind of looked at this in a larger study. It was 4.2%, and the prevalence of mitochondrial disease from our experience within mitochondrial disease is that if you take this whole spectrum of mitochondrial disease, it's only the last quartile, and really the last 10 to 20% of the children who have the mildest forms of mitochondrial disease that can even, be meaningfully tested to meet criteria for autism spectrum disorder. And I'll tell you that the other parts, of the children that have the even more severe multi-organ, dysfunction, yes, language delay is an issue, but that's a higher order function that is common in a lot of different neurocognitive abnormalities. But their socialization is actually very good. Okay, so it's not so. The majority of mitochondrial children do not have social engagement abnormalities. But, okay, so getting back to this. So it turns out that if we say that 4.2%, that 4.2% of 1,000 is 42 kids, but we already know there are only 25 children out of 100,000 that have mitochondrial disease. Okay, so it's just mathematically impossible. So how do you resolve that? Well, you can reduce the prevalence of real definite mitochondrial disease within autism, and this is a study that came out recently by Corrigan and Dager that looked at proton MRS in order to look for characteristics of mitochondrial dysfunction in the brains of children, 54 children with autism. And in those cases, basically zero of the kids had characteristics that were associated with primary mitochondrial disease. Now, some kids did have elevated lactates in their brain, but it wasn't any different from the controls. Okay, all right. So now, how do we come to grips with this? So let's keep the prevalence of autism spectrum at 1%, but let's lower the prevalence of mitochondrial disease within autism. So, and again, this is definite genetic forms of mitochondrial disease, let's keep the prevalence of autism spectrum disorder as an endophenotype within mitochondrial disease at close to 10 to 20%. Now, when you do that, and let's also change. Let's lower the rigor with which we identify definite mitochondrial disease, and call this probable or possible mitochondrial disease. And now, what you do is we actually come to something that's a little more compatible. So we have 1,000 children out of 100,000 that have autism. Let's say that 1% of those have mitochondrial disease, and then 40 children will have mitochondrial disease without autism. And so you end up with a total of 50 children out of 100,000 with possible or probable mitochondrial disease. Okay. All right. So this is just another way of saying it, that if you look at mitochondrial disease and you look at their overlap with autism spectrum, we have something on the order of 1 to 4%. And 96 to 99% do not have primary mitochondrial disease, but they could, they do have secondary, and many of them have secondary forms of mitochondrial dysfunction as associated as a manifestation of this principle of metabolic matching that mitochondria, orchestrate. Okay, so, that's the exposure to the environment. That's their bioactive chemical exposures in the environment, as well as genetic predispositions. All right, so it's important to really recognize this, because children with autism have a different response than children with mitochondrial disease to a variety of different interventions. So valproate, for example, reliably produces deterioration when used to treat seizures and most mitochondrial disease, but 71% of children with ASD improved in a paper by Hollander. Fever will reliably produce deterioration in patients with mitochondrial disease, but in a study by Curran, about 50 to 80% actually had improved language and social engagement in children with autism and fever. Hyperbaric oxygen reliably produces either no effect or deterioration in children with primary forms of mitochondrial disease, but 30 to 80% improved according to a paper by Rossignol. Spontaneous recovery, as you might imagine, of genetic forms of mitochondrial disease is extremely rare. But in autism really is a treatable disease, and the quoted numbers for recovery are 3 to 25%. So it's actually good to be able to have a secondary dysfunction disorder that you can potentially treat by eliminating the environmental trigger and knowing more about the kind of individualized, personalized response of each child to their environment. Okay. I'll go through this quickly because I'm running down to 5 minutes of slides, and I wanted to give you a little bit of ecogenetics at the end. But I want to emphasize the point, for those of you who haven't seen this talk before, is that not only every tissue have different mitochondria for that tissue, but every tissue has a different number of mitochondrial DNAs that determine the work capacity. So in this first line, we talk with a secondary oocyte of, and I say 0.9, but it's 0.2 to 2 million mitochondrial DNAs. And then what happens in the embryo? Okay, you start out with that, say 2 million mitochondrial DNAs, and in the zygote. In the male, the contribution to actually about 50 mitochondrial DNAs brought in by the sperm, but they are killed after the 8-cell stage of development. So you're only left with the mitochondrial DNA of your mother. Okay, average unrelated people here will have about 30 to 50 different, differences in their mitochondrial DNA, but you'll all be clones of your siblings that are, through which, you share a mother. Okay. Anyway, so as you start dividing, you actually have non-replicative divisions of mitochondrial DNA. So you go from, let's say, 1 million to 500,000 to 250,000 mitochondrial DNAs. And the very first tissues that start appearing and taking form, in the growing embryo are the ones that actually do have the highest numbers of mitochondrial DNA contents, like the heart and the central nervous system. Okay, so cardiac muscle has about 6,000, CA1 hippocampal neurons about 6,000, quadriceps skeletal muscle about 3,000, adipose 2,000, white blood cells about 400, and skin fibroblasts about 100. Okay, leave it at that. There's a concept of heteroplasmy it's important to know. Not all your mitochondrial DNAs have to be identical. You can have point mutations in mitochondrial DNA. Our repair mechanism, so normally you're born with what's a condition of homoplasmy, which is 100% of your mitochondrial DNAs are wild type or normal, okay? But there are children that are born with point mutations that affect some mitochondrial DNAs but not other. So in this case of 4 mitochondrial DNAs affected out of 10, that's 40% heteroplasmy. As you age, it turns out that our error repair mechanisms after about age 50 are able to keep, before age 50 they keep track of somatic mutations, are able to maintain corrected mitochondrial DNA damage, but after about age 50 you start accumulating mitochondrial DNA point mutations and deletions exponentially until you die. It doesn't stop. Okay. All right. So there are over 200 different point mutations that cause mitochondrial disease. We discovered was the first point mutation in mitochondrial DNA. It's this lysine tRNA mutation that's associated with autism in a family. So this is that family. We published this in 2000. You can see the mother in blue here. She had a child that presented to us, a little girl, in that circle over here, that had a profound lethal form of mitochondrial disease called Lee syndrome. We investigated her brother who had learning disabilities, who actually had a regressive disease that was diagnosed as autism. And so this was the first case where where a single mutation could be associated with, on one case, high levels of heteroplasmy, 82% in the female, leading to Lee syndrome, and lower levels of 60% in the blood, leading to autism in the child. And the other point that I want to bring out, okay, so the girl had decreased complex 4 and 5, but the boy, even though he had the same mutation, he actually had increases in his complex 1 respiratory chain activity. So overactive respiratory chain activity in response to, basically as a compensatory response to a mitochondrial DNA mutation in this case. You can also have this overactivity in response to non-genetic mitochondrial insults. Okay, so I'll leave it at that. I think I'm just going to whiz through the rest and get to, so part of the point of this afternoon's talk is really to try to coordinate and emphasize the connections between our groups. And immunomitochondrial biology is what I call the secret life of mitochondria. I mentioned already that I believe that the cellular defense function of mitochondria are more ancient than the energetic functions of mitochondria. And as said by John Dryden in 1681, self-defense is nature's oldest law. If you don't survive, then you don't pass anything on to the next generation. So mitochondria control the first steps in innate immunity. There's a mitochondrial antiviral sensing protein that interfaces with the RIG1 helicase, which actually binds to double-strand RNA and DNA, and that coordinates a phosphorylation reaction that eventually results in activating the interferon response factors 3 and 7 that then go to the nucleus and coordinate interferon response of that cell. Okay, this is also involved in NF-kappa B activation. It's not obligatory for NF-kappa B, but it amplifies the NF-kappa B response. Okay. Off to the right, I have circulating, so MAVS is the mitochondrial antiviral sensing protein. So off to the right, I have an article that talks about mitochondrial DNA and mitochondrial formulated peptides as being damage-associated molecular patterns when they are released from the cell that are pro-inflammatory, and I'll give you an idea what that actually means. So when damaged mitochondria are released, their DNA and protein fragments are chemotactic. They actually call in neutrophils, call in phagocytes, activate phagocytosis. So on the left is a pipette that contains mitochondrial DNA, and you can actually see the neutrophils chemotaxing to the tip of the pipette. So I'll show that a few times on the left. And then on the right, you have a pipette that's filled with the same mitochondrial DNA, but now the same neutrophils. But now we've added a protein, an antibody to the alpha formal peptide receptor, the formulated methionine, the peptide receptor, and you can see that the neutrophils just kind of hang out and talk to one another in coffee clatches. They don't do the same thing as they're doing over here. So over here, they're going right for the tip. And on the right, they're just kind of hanging out. Okay. So, all right. So we're running low on time. But I'll point out that there is a kinetics of response that primary mitochondrial disease has in response to infection that is different than when children with autism spectrum disorders, will have regressive events. It's frequently associated right with you'll have a very exaggerated, response at the time of peak fever, but in mitochondrial disease, you'll have it as a delayed symptom where there's a fade response that occurs 5 to 10 days after the peak of symptoms. Okay, where a child will stop talking, stop walking, stop being able to swallow. They'll have a stroke-like episode. It's a fade response, and that's from bioenergetic failure, because the real need for mitochondria really kicks in. Remember, I told you that mitochondria actually dial down with acute injury. Okay, so it turns out they dial back up, during recovery and regeneration. That's essential for healing. Okay, and that regenerative phase is the phase where we really start seeing the biggest metabolic stress on the kids with primary forms of mitochondrial disease, and that's where they really start cascading into problems. There's also a flare response that occurs coincident with the peak of seizures or peak of infection inflammatory response. At between 1 and 3 days, and that's due to an exaggerated immune response, and that's something that was characteristic of the Hannah Pauling case that brought really mitochondria and autism together in the newspapers in 2008. Then there's also something called a storm that occurs really within the first few hours, that can extend for days, that occurs with as a memory response or anamnestic response, and that's something that occurred in the very first patient subject of gene therapy was Jesse Gelsinger, who had OTC deficiency. He was given the OTC gene and the adenovirus vector. It turns out he was previously immune to the adenovirus vector, which is a relatively common cold virus. And he developed disseminated intravascular coagulation, shock, and died. Okay, finally on to something that I really wanted everybody to be awake for. So anyway, this is equigenetics. It's the study of genes that interact with environmental factors to lead to health and disease, and it's also the study of the environmental factors that interact with those genes. Now, So remember, autism is kind of interesting. It's a moving target. If you go back and you plot the prevalence of autism dating back to 1990, and you can actually create a very nice exponential curve that goes through about 2005, with 18% growth per year. Okay, and then it starts falling off a bit. Okay, it starts falling off a bit from there. And so whether that means that we're kind of coming to a new equilibrium with our environment, or our environment is getting better, or what's happening, it's hard to say. But I've been told, and the next slide tells me this, from Autism Research Institute statistics, that 80% of the patients in the U.S. with autism spectrum disorders are under the age of 22. Okay. And if there anybody with better statistics than that, I asked them to come tell me to correct this, but this is the number that I have, and it's really kind of a shocking number. Okay. So what about the ecogenetics of mitochondrial DNA? So we evolved in Africa, and then our ancestors moved to the Middle East and Northern Europe and Asia, and crossed across the Bering Sea into the Americas. That all occurred tens of thousands of years ago. Okay, but our ancestors dwelled, like Moses will dwell in this land. We dwelled in a land. Our ancestors dwelled and became part of that land. A thread in the web of Chief Seattle and Chief Seattle's web. We learned to live with the nutrients in that environment. We learned to survive in the presence of the different microbial environment. In the cases where there was malaria, we developed resistance to malaria, and where tuberculosis was common, we developed resistance to tuberculosis. So, but what happened recently in the last 300 years is that we had this massive exodus from Northern Europe and from Africa to the North America, and that brought in genes that had all co-evolved with their environments, for thousands of years earlier, into a new environment with new food sources and new viruses and bacteria and fungi to evolve with. And then, just as we get good at that, then we change the game again and start, with dramatic changes in the way that we do farming, in the way that we do. That we do our fishing, where concentrated animal farming operations, really are producing more and more of our food supply, and we're, using the powers of our brain to generate chemicals that we've, that our environmental history, our evolutionary history has never encountered before. Okay, and so we're having to readapt, okay, to this changing environment. Something that's really interesting that's put this in sharp relief for me is I do some collaboration with investigators in India, and India is dominated by this so-called M haplogroup of mitochondrial DNA. But before about 30 years ago, so in fact as little as 20 years ago, when I first went to Bombay and just was sitting in the airport, fewer than 10% of the people were obese. Last year I went there and 30% of the people in the airport were obese. Now it turns out there's an epidemic of diabetes and cardiovascular disease in India. It's also happening in China. This is ongoing ecogenetic disease that is occurring because our environment is changing faster than our genes. Okay, and I'll bring this home for autism in particular. So with the political unrest in Somalia, a number of refugees went to Kenya in 1993. And in 2005, a large population, a large population immigrated to Minneapolis, St. Paul, in the mid and late 1990s. The prevalence of autism in children in that original Kenyan population or from Somalia was less than 1 in 1,000. But in 2005, the prevalence of autism in Somali Americans, first generation Somali Americans, was 1 in 60. So the statistics were actually kind of interesting. So in 2005, there was a prevalence of autism among Caucasians and Hispanics that's a little bit, significantly lower than we talk about now as 1%. It was 0.17, and at that time it was just 0.28%. And it rose to 0.28% by 2008. But 1 to 1.5% of the first generation Somali Americans, had autism, and that started falling back down to in 2008. So this is all statistics from the Minnesota Department of Health that you can actually access on the web. So if you look at this, this is another example of the moving target of autism and this ecogenetic adaptation that's occurring. So in the beginning, the prevalence of autism among Somali Americans compared to non-Somalis was nearly 7 times greater than sevenfold greater. In 2006, it was 5-fold greater, and in 2007, it was 3-fold greater. Okay, so there's an adaptation that's occurring, before our very eyes here, as this new genetic population is adapting to their environment. Okay, that's it. Thank you very much.
The Autism Research Institute depends on the generosity of its donors, like you, to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.